In a previous lesson on the role of the hippocampus in memory processing, we learned that the hippocampus can do two important things, what we call pattern separation. The hippocampus has the ability to assign distinct events to different populations of cells to make non-overlapping memory traces, and then also pattern completion, that when a cue, one aspect of an event, is represented, it can reactivate the hippocampal memory trace, which then can reactivate the cortical memory trace. So we call that pattern completion. In this lesson, we'll take a look at some research that supports this idea that retrieving a memory is going to be reactivating some of the cortical cells that were involved in processing the event in the first place. Now, the research will involve recording the electrical activity of single cells. Here we see the left side of the brain, and this region of brain down here is called the inferior temporal lobe here. And if we turn the brain sort of upside down so we can see the bottom surface, the outer part of the underside of the temporal lobe would be a visual area, a high-level visual area. And then as you go towards the middle part of the brain, that surface is part of the medial temporal lobe, and that's more associated with memory systems, because underneath this temporal lobe is the hippocampus. And so what researchers did was they inserted recording electrodes, and they found cells in the visual region here, and also other cells in the memory, the medial temporal lobe, the memory part of the temporal lobe, and they were able to record their electrical activity. So when the animal sees some visual stimulus, they would find cells in the visual area that responded to that visual stimulus. And then what they had the animal do is try to remember an association between one visual stimulus and another visual stimulus. And so they wanted to find cells that were activated for the first visual stimulus, cells that were activated for the second visual stimulus, and then they were going to examine how the cells behaved during retrieval of the memory of that association. So it's a paired association task. Animals learn to associate certain visual cues with certain other visual stimuli. And again, they're going to be recording single cell activity in perceptual areas and also the memory areas. After training, researchers present the cue and measure brain activity as the animal retrieves the memory of the associate. And the results are going to support the reactivation hypothesis that memory retrieval reactivates brain areas used in the perception of the stimulus. In fact, reactivates brain cells. They're recording from single cells in this case. Okay, so here's how the sort of experiment proceeded. First, at this phase of the experiment, a visual cue is given to the animal, and the researchers had to find cells that responded to the visual cue. So here they found the red one here is in the visual perception area, area TE. We're going to call that a TE neuron. And then the green one here would be a cell in the sort of memory region of the temporal lobe. We'll call that MTL neuron, medial temporal lobe neuron. And the idea is that we've already learned in past lessons on the hippocampus that visual information is fed into the hippocampus as part of the process of forming an episodic memory. So we have the flow of information. This visual neuron is responding to the visual cue, and it's relaying information to the medial temporal lobe neuron, and that's going to send information down into the hippocampus, and hippocampal neurons will fire, etc. So this is processing the cue stimulus. Now, if you record the activity of these neurons, there's the TE neuron, the visual stimulus, the visual neuron, is activated a little bit before the MTL neuron, and that makes sense with this flow of information. The visual information is going to activate the visual neuron first, and then it's going to activate the MTL neuron, so you get a little bit of earlier activity for the visual neuron compared to the MTL neuron. So that makes sense. Now, during the training period then, the animal is exposed to the visual cue and its paired associate. Now, because the paired associate is a different visual stimulus, a different set of cells will be activated. So here the blue neurons are going to be activated in the visual part of the temporal lobe and the memory part of the temporal lobe. But again, that information will find its way down into the hippocampus, and the hippocampus, because these visual stimuli are being paired, the hippocampus is going to be linking those two memory traces. So we're going to get LTP changes down here, synaptic plasticity. The hippocampus is going to be linking those two visual stimuli because they were presented together. So the cue is linked to the paired associate. So the interesting question then was, what's going to happen during retrieval? So if the animal that has already now made this association between the visual cue and the paired associate, what's going to happen if the paired associate is presented? Will the same cells that were involved in processing the cue be reactivated as the memory is retrieved, the memory that the cue was paired with the associate? So that was the question. They already found the red and the green cell that were active in processing the cue. Would those same cells be reactivated when the animal was retrieving a memory, the memory that a certain cue was paired to an associate? So would we get this kind of information flow going in the backward direction? Would the paired associate reactivate the cue memory, which would then activate the red and the green cell in a different kind of sequential order? Would the green one be active before the red one? Well, when they gave the animal the paired associate after training, sure enough, they found that the green cell, the MTL cell, fired before the red cell. So both of the cells were active and the temporal order activity was reversed. So here we see the record down here. The MTL cell fires first, there was a delay, and then they found activity in the red TE neuron. It was as if the information flow was going in the backward direction. So the paired associate had reactivated the memory trace for the original cue stimulus, and that involved cortical reactivation of the MTL cell followed by the TE neuron. So here was evidence then that retrieving a memory is activating the very same cells that were involved in processing that stimulus in the first place. With our graph down here, the idea is that the MTL neuron's activity was the trigger for the activity of the TE neuron, which showed the delay here. So the TE neuron's activity is part of retrieving the memory for the cue stimulus, and this supports the reactivation hypothesis that memories are in part stored in the same population of cells that are used to process the stimulus in the first place.